I'd now like to introduce Dr. Larkin of the University of Florida, and he will be giving us a talk on the impact of lunar radiation gravity on plant growth and rhizobium communities. So, Emmerich, it's all yours. Thank you.
Hey, I'm a PhD candidate at the University of Florida. This talk was co-authored by Dr. Melanie Carell, whose lab this was conducted in. Today, I'm going to be talking to you about investigating the impact of altered environments on lettuce rhizo communities. So not quite rhizobia. Next slide, please. So a plant microbiome is an ecological community of microbes. Those could be bacteria, fungi, viruses, even microprotozoa that are associated with a plant host living in or on its cells. Next slide, please. And particularly, we're going to be talking about a certain compartment of that plant microbiome called the rhizosphere. And these are microbes that are closely associated with the root systems of plants. Next slide, please. The lettuce rhizosphere, like other rhizospheres, can harbor a diverse range of microbes that can either be relatively neutral or commensal with the plant, can have direct growth promoting properties that produce hormones that augment the way the plant grows or help it deal with stresses. And it can also harbor plant pathogens that overtly cause infection in the plants. All of these are relevant to formulating strategies for managing agricultural systems, understanding how all of these interact with other parts of the microbiome. And in controlled environments, particularly, we're concerned with human-associated microbes that may or may not have a strong effect on the plant themselves, but we're concerned about them because of their effect on us. And these can be things like opportunistic pathogens. A lot of the microbes that are present in recirculating built environments are human-associated, especially on places like the space station, where humans are the source of a lot of the microbes in the air and on the surfaces. And we want to understand how these microbes persist on the leaves and in the roots of any plants that we may or may not be consuming. Next slide, please. So in these controlled environments, we can provide plants with more or less optimal conditions for growth. This is, a cornerstone of life support concepts using plants, but researching these kinds of systems is also important for the expanding field of things like urban and vertical farming, and also for researching field agriculture. Next slide, please. So what kinds of gaps in understanding do we have in the microbiome as it pertains to these controlled environments? Well, although we can control the environment to a certain degree, providing certain gas mixes, different kinds of lighting and nutrient regimes, we still have a very limited understanding of how environmental factors and environmental perturbations affect these microbial communities and even their hosts as well. We need to understand how host genetics impact microbiomes. We know that even cultivar level differences between the same species of a plant can have significant impacts on the composition of a microbiome. And we need to understand this interplay between the host ecological factors and the microbial community themselves. On a surface that's not our own, like the lunar surface with all these new environmental components, we know even less. And we need to understand the impacts of things like cosmic radiation and altered gravity if we want to employ strategies that take advantage of these microbiomes that have such a strong relationship with plants for more sustainable and stable agricultural production. Next slide. We've been building a picture of what's regular, what do we find recurringly in these lettuce rhizospheres, because that's an important precursor to developing agricultural strategies that use these microbes. We need to understand what persists and what has sort of an ecological niche that makes sense in, from host to host and management system to management system. We're looking at several different hydroponic lettuce production experiments. Some of them looking at things like the use of urine-derived fertilizers for life support systems like the Melissa system, and other more standard NFT systems with standard nutrient management practices. We're also looking at some field lettuce, particularly romaine cultivars that are very phosphate efficient, phosphate use efficient. We're also looking at lettuce, particularly outrageous, from some NASA experiments that were grown on the space station with ground analogs. And we're also looking at the use of some commercial inocula that have been developed to promote the growth of leafy greens in controlled environment settings. And then hopefully in the future, we'll be using what we've learned here as a lens to understand what kinds of effects the reduced gravity and increased radiation exposure that the lunar environment will have on both the plant and the microbiome. Next slide. These are results from the urine-derived fertilizer experiment. On the left, you can see a very complex network model from the samples that were grown using a particular urine derivative that produced very poor results. These plants were very salt-stressed, did not produce marketable lettuce. And then using the same network modeling techniques, the same statistical thresholds, you can see networks produced from the best performing treatments, that being the control, a Hoagland solution, and a K-struvite-based urine derivative. These networks are much less complex, and they have a more positive interactions to negative interaction ratio, and in general are more similar to each other than they are to the more complex network from the ED preparation. Next slide, please. Apart from network structure, that same concept is reinforced. You can see here on this ordination of the Bray-Curtis beta diversity index, the orange and green samples are coming from that control Hoagland solution and K-struvite solution, and they're overlapping quite closely on top of each other, showing the similarity of those communities. Next slide, please. Here's a phylogenetic composition of the bacterial rhizo communities from the field lettuce experiment, looking at the different rhizo compartments. Next slide, please. And the same concept for the fungal communities, in this case using the ITS marker gene for the same experiment. Next slide, please. On the left, you can see outrageous lettuce. This is a lettuce that has been commonly grown in the life support context research-wise. And this lettuce was grown with a commercial inocula from the company Synergy. On the right, you can see the nutrient solution of the hydroponic system a few days after that inocula was put into the system. There were several different inocula tested here. They all produced good, healthy lettuce crops, and we're still analyzing that. Next slide, please. And so far, we've learned that the bacterial communities in these lettuce rhizospheres are dominated by particular phyla across environments and management practices, which bodes well for the development of strategies that utilize these microbes for boosting those agricultural systems in sustainable ways. The same has generally held true for the fungal communities, though there's lower amount of dominant phyla, and that dominance isn't quite as severe, although Ascomycota does break that rule. And in general, the rhizosphere communities that we've seen, like in field agriculture, are less diverse than the nutrient solutions in which they are growing in, similar to the way that the bulk soil is more diverse than the rhizosphere in field conditions. And in all of the studies we've looked at, any management practices or treatments applied have had strong effects on the microbiome. Next slide, please. Moving forward, we'll be continuing to aggregate these data sets and looking at them in different ways. We're also including some additional experiments and are actually looking for other interesting lettuce systems to sample. And we'll build a more robust picture as we add more experiments into the data set of what do we recurringly see in these lettuce rhizobiomes, and what seems to be a common theme across these different environments and practices. On the left, you can see a Venn diagram of the core microbiome and across the different interactions of of these treatment groups in the urine derivative study, and you can imagine a similar diagram where the lobes, instead of being treatment groups, could be different types of hydroponic systems, aeroponic versus NFT, or different types of environmental factors such as partial gravity, full gravity, microgravity, etc. Next slide, please. Moving forward, we want to move away from just sort of building a survey type picture of who's there and how is that community changing over time, towards understanding what these microbial players are doing or what they have the capacity to do. And in particularly, we would like to understand what kinds of functions could enhance plant growth, protect against environmental stresses like gravity and radiation exposure. A great way to investigate this in the context of cosmic radiation exposure would be to have parallel lunar, Earth, and space station experiments using growth chambers that can match environmental conditions beyond the altered radiation and gravity environment. And this should be feasible, seen as many sequencers have seen use on the space station. There's still going to be issues with tissue processing, and this kind of experiment will likely need to be automated, but could be done with maybe minimal crew involvement. There's going to be engineering hurdles here. I don't have any specific propositions on how to make that happen, but I'm sure that we can figure it out. And in general, I just have an intuition that the long-term effects of cosmic radiation is going to be more impactful and a bigger hurdle for long-term agriculture operations on lunar planetary surfaces. So I think we should pay some more attention to that. Next slide, please. And I think a great place for this to happen would be the Shackleton crater, where near constant light is provided. And if we can set up the right infrastructure, there will be energy and water accessible. And we can set up something like an advanced plant habitat, which already exists on the space station and on Earth, where we can then set up plant growth experiments that also incorporate microbiome sampling, where the environmental factors provided are essentially identical, with the exception of those things that we can't control, such as gravity and radiation. And we should be able to start singling out those factors and understanding how they affect the microbiome, so that we can then help develop strategies and/or use strategies that we've discovered on leveraging the microbiome for safer and more productive agriculture systems. Next slide, please. And that's it. If anybody has any, I know I didn't go into specifics of a lot of things, just wanted to cover all the things. But if you have any specifics on some of the slides that I went over, feel free to email me. Next slide, please. Or post it in the chat. And I'd like to give thanks to my PhD committee, professors that I've collaborated with both at UF and abroad, graduate and undergraduate students that have helped me along the way, and my funding sources, the McKnight Foundation, the Florida Space Grant Consortium, and the USDA. And that's it. Thanks.
Great. Thank you, Eric, for that wonderful talk on a very important area of science that we certainly in space biology and NASA are moving into, which is the microbiome, and also the great part on the interaction in communities with the rhizosphere community.